Hello, everyone. Welcome. For those of you that don't know me, I'm Corey Moreau, and I've had the honor of being Benjamin Blanchard's thesis advisor. I know Benjamin both academically, also as friends and as family, so we're really thrilled that you could be here today. Before we begin, I want to take a moment to explain some of the logistics regarding this online platform, since this is unique. And my first video recorded thesis defense, and I suspect for many people it's theirs as well. First, we have made sure to mute all of you, and if at some point you notice that you're not muted, please mute yourself. Also, please make sure that you turn your videos off during the thesis presentation to help increase the probability that we have a smooth video stream throughout this. As you likely heard, this presentation is being recorded, and Benjamin will share that in some way in the future. We're going to ask that you hold all your questions and comments till the end of the presentation, at which time we will be able to entertain questions. And during the question period, there will be several of us that can actually help moderate the questions. If you do have a question during the presentation, you can add it to the chat conversation found along the options on the bottom. I'd also like to ask everyone participants button on the bottom at this time. This should bring up a window on the right side of the list with all of the meeting attendees, and you'll see an option to raise your hand. So you can use that once we've started the question section so that you can alert us that you actually have a question. But please remember to unraise your hand after you've sort of follow along in the queue with people who have questions. So these questions for Benjamin will come from the chat section as well as the virtually raising your hand, and we will unmute you so that you can ask your question. Directly following the oral public thesis defense and question period, Benjamin will then meet with just his thesis committee to further discuss his thesis research. After this, and assuming there's been a positive outcome from today's events, we will virtually celebrate with Benjamin using another Zoom video conference room. I'll paste this URL and password into the chat window shortly. So be sure to copy it down somewhere and log in about 1 hour. And as soon as we're all finished with the private portion of the defense, we'll join you. So now that I've addressed all of the housekeeping points of this virtual thesis defense, I'm thrilled and honored to be here today to introduce Benjamin Blanchard. I first met Benjamin when he came to interview for graduate school when I was still at the Field Museum in Chicago. And although Benjamin's from Michigan and completed his undergrad degree at the University of Michigan, he actually flew from China for the interview. Was there teaching English for a year and sharpening his Mandarin language skills, which he's been working on since high school. Even jet-lagged and tired, Benjamin spoke faster than anyone the fastest talking person in a room. There are many qualities about Benjamin that make him stand out, but his passion for ants is among the ones that I admire most. I remember that his application essay for graduate school literally started with the sentence, "I love ants!" exclamation point. So most students actually wait until they start graduate school to begin actively trying to figure out what their thesis research will be, but not Benjamin. I remember he emailed me several times, months before beginning graduate school, running ideas by me and sharing aspects of ant biology he found interesting. After a few of these emails back and forth, I sent him an idea I had, and I'll quote the text of my email to Benjamin. So this email is dated August 19th, 2014, and here's what I wrote to him. One observation that I've been knocking around in my head and may not actually have data to support is that my overall impression in the ants with heavy spinescence, Polyrhacus, Phytolus, Cephalotes, do not have other forms of defense well developed, the sting, venom, mandibles, etc. I'm not sure if there's a trade-off or if making one or the other is energetically costly. It may be interesting for your first project to do a comparative study across the ant phylogeny to look at correlation of traits related to defense, and maybe diet. Do predators have more highly integrated defense characters or other functional phenotypes? I wanted to read this quote directly because, as you'll see during Benjamin's defense, he took this idea that I had and ran with it. Not only did he show that some of my intuition was justified, he went far and above and produced a very rigorous study that was published in Evolution in 2017, and it's already been cited over 30 times. In addition, Benjamin was awarded second place in the student competition at the International Congress of Entomology when he presented this work. Another aspect of Benjamin's research that really stands out is that he has conducted meticulous and elegant research that spans evolution, comparative biology, field-based ecology experiments, and bioinformatics. Research in any one of these fields would have been enough for a dissertation. For his field-based ecological experiments, Benjamin spent 3 summers in China, funded by 2 separate National Science Foundation grants that were to Benjamin himself. Although it may not seem obvious from Benjamin's presentation of these experiments required many hours in the field to collect the colonies across multiple species. In addition, the experiments themselves took many, many hours of recording data. All this work paid off, and this manuscript was just accepted this morning, no kidding, in Ecology and Evolution. To date, Benjamin has authored or co-authored 9 peer-reviewed publications. In addition to his research, Benjamin is committed to scientific outreach. He has volunteered to judge local science fairs and served as a volunteer academic tutor for youth from Chicago's South Side. He has participated in many outreach events at the museum and even hosts an online blog called The Daily Ant, which you should all check out if you're not already following. On a more personal level, Benjamin is he can transition easily from talking about his or other people's scientific research in a very sophisticated way to sharing ant facts that are fun and digestible to any level of audience. I have really enjoyed interacting with Benjamin over these last 6 years, and I'm happy with the work he has produced, and I'm proud to call him a colleague. Although Benjamin is not certain what he'll do next, he has 2 very prestigious postdoctoral fellowship applications pending. One thing I know for certain is that no matter what Benjamin does next, he will bring excitement, passion, and intellect to it, and is certain to be highly successful. And just a final reminder to join us 1 hour after Benjamin's defense in the video chat that I will paste in the chat window below to congratulate Benjamin pending a successful thesis defense with this committee. So without further ado, I'll turn it over to Benjamin to share the outcomes of his excellent thesis research. Take it away, Benjamin.
Thank you so much, Corey, for that extremely gracious and heartfelt introduction. I am going to now change to share screen. Hopefully this will work fine. Okay, good. There we go. Slideshow, play from start. Corey, actually, if you could unmute for a second just to confirm that that you can see the.
It's working perfectly.
Awesome, great. Okay, cool. So let me minimize this. Great. So thank you so much, everyone, for tuning in. It really means a lot to me, and I really appreciate all of your electronic presence here for my talk. Today I'll be talking about the evolution and ecology of defensive traits in ants. I know it's a big shock that I study ants for all of you, but one of the motivating reasons why I'm interested in ants and why I am interested in defensive traits is that ants are extremely dominant across the world. I would bet that all of you have encountered ants before, and there's over 13,000 known species in this group, and they're also so they exhibit a variety that is really pretty stunning. And just a very small snippet of that variety is displayed here, which is showcasing some defensive traits, where we can see a chemical stinger, some worker polymorphism, where you have soldiers and miner workers. You also have some spine morphological defenses, some behavioral defenses with large colony sizes and mandibles. And again, this is just a small snippet of the total variation in this group. And if you zoom in into just one species, which you can see here, there's a number of questions that can arise and which evolutionary biologists and ecologists are interested in asking. And in fact, these fit pretty well into the 5 W's, which some of you may remember early English classes if you studied the English in the US at least. The first of these is who. Who is this species? In this case, this is the red imported fire ant. And some of you may have encountered the joys of this ant before. You may ask, what is the behaviors that it's doing? What are the traits that it has? Here, it's biting onto the hand of the photographer, Alex Wilde, in order to sting him more vigorously, and so it's using a stinger. You may ask, where are these ants distributed? In this case, this is an invasive species, and so you might be interested in its invasive range as well as its native distribution. You can also ask, when did the species evolve, and when did the traits that did evolve? And here we would be talking on an evolutionary timescale of millions or tens of millions of years. And you may ask, why did these traits evolve? In particular, what sort of ecological processes might have driven the evolution of these traits in your group of interest? And so it's kind of interesting to me that these 5 Ws work not just in English class, but also in evolutionary biology as well. And I sort of gather these all of these types of questions into a single motivating question, which is, what are the ecological causes and evolutionary consequences of morphological defensive trait adaptations? And here I'm interested in trying to understand what has promoted both the ecological and evolutionary success of ants. My dissertation consists of 3 chapters, the first of which is on defensive traits, assessing evidence for an evolutionary trade-off and evidence for trait-based diversification, and that's broadly across all ants. The second, for the second chapter, as well as the third chapter, I zoom in into the beautiful spiny ant genus Polyrhacus and investigate the role of spines on various ecological traits in this group. And for the third chapter, I use new methods of phylogenomics to assess trait evolution in Polyrhacus and its potential impacts on diversification and geographic range size. So to move in on the first chapter, there are a number of studies that have found an association between defensive traits and diversification. And these have occurred across many different taxa that are quite disparate, including chemical defenses in poison dart frogs, ant-mediated extrafloral nectary defenses in plants, and a hind leg jumping morphology in beetles, among many, many other examples. This suggests that defensive traits are important to investigate if you're interested in trying to understand why species have diversified as they have. Similar to trait-based diversification, the concept of a trade-off is very common in both the ecological and evolutionary biological literature. And this concept is simply that any one organism or species is not able to express all possible traits that might be well adapted to all possible selection pressures in its environment, and so there must be some trade-off in traits. And the concept of a trade-off is probably one of the most fundamental in both ecology and evolutionary biology, and it dates back to Darwin and earlier. What you can see in this example here is with Darwin's finches, where there's a trade-off between bite force and, between bite force and jaw closing velocity, which is mediated based off the food resources, the seed resources that are being predated upon by these birds. In ants, if you look at the genus level across all ants, there's an apparent kind of qualitative appearance of a trade-off where in defensive traits, where it's rare, for example, to have a genus that exhibits some large eye size, a stinger, and also worker polymorphism. And so it's possible that there's some kind of trade-off going on. But whether or not there is an evolutionary trade-off, and in fact, really any understanding of a trait-based diversification in ants with defensive traits or otherwise has largely been unstudied in this group. And so this study was focusing on trying to fill that gap. So overall for this project, I have 2 core questions. The first is there an evolutionary trade-off between different defensive traits in ants? And the second is, how does the evolution of defensive traits impact clade diversification? In order to address this question, I first compiled a large trait database of a number of traits which I binned into binary discrete categories. And the 5 morphological traits that I used have at least a partially defensive function in the genera in which they occur, or often have a defensive function, although I recognize that for some traits there can be other functions as well. One example would be worker polymorphism, where you can have the worker caste is divided into different morphotypes, where in many cases you have a soldier major worker and a minor worker, where the soldiers are at least in part defending the nest from any attackers. So I binned all these categories into, for colony size, this was small and large colony size, small and large eye size, monomorphic or polymorphic, and the presence or absence of cuticular spines and a chemical stinger. And I gathered these data from existing information in the literature, as well as online databases like AntWiki and AntWeb. And I also collected some information on ecological traits, but I won't be talking about those results today. This database included every ant genus, at least what was every ant genus at the time. Taxonomy moves very quickly, so some that has shifted already. And it was therefore a pretty large data set. In order to maximize the use of these data, I also inferred a phylogeny of the ants, a genus level phylogeny. And this was using molecular data sets from 6 studies, which included 9 gene markers. And I first inferred a maximum likelihood topology using maximum likelihood inference in the program XML on the Cypress computing cluster. I then used the resulting topology as a starting tree for Bayesian inference and dating analyses in BEAST using 73 fossil calibrations. And these included 271 in-group genera. And from this, I inferred a maximum clade credibility tree, which was used for downstream analyses. Combining the trait database with the phylogeny, I then conducted a number of analyses in order to get at both the questions of trait-based diversification, as well as assessing evidence for a trade-off. And the first of these were sister clade analyses, which is a long-standing method where what you do is you, across the tree, select pairs of clades where it's true that all of the descendants of one clade have one trait state in a binary trait state, a trait. And all of the descendants of another trait have a different trait state. So here this could be 0 and 1, potentially small colony size and large colony size. And because you do this throughout the tree, you actually get somewhat phylogenetically independent comparisons where you can then compare the diversity between these two clades for each pair. And also the specific implementation that I included also incorporates the time since divergence as well. It's a slightly model-based approach. I also implemented a rate shift analysis in the program Medusa, which is useful for assessing, without incorporating any trait information, where there might be shifts in the diversification across your phylogeny. And I won't talk about this in too much detail, but I will reference the results for the shift analysis later. Then, in order to address these questions about trait trade-offs, I also conducted a maximum likelihood test in the R package quorum. And this is a model comparison method, and it's a little bit confusing at first, so I'll walk through it. This compares an independent model of evolution between two binary traits to a dependent model of evolution. In the independent model, you assign two parameters for the gain or loss of one trait, like large eyes, the gain or loss of large eyes. And you assign two parameters for the gain or loss of a second trait, let's say the chemical stinger. And what's notable here is that the gain or loss of any trait is not dependent on the trait state of the other trait. The gain or loss of large eyes has nothing to do in the model with the gain or loss of a stinger. If this model is favored, then there's no evidence for any kind of a positive or negative correlation, no evidence of a trade-off in the negative correlation case. But in the dependent model, there's 8 parameters assigned. And now here, there is a model-based way of incorporating information such that the transition for one trait is in fact based on the state of a second trait. For example, here we see that the gain of large eyes in the absence of a stinger is given a different arrow, a different parameter, shown here in dark blue, from the gain of large eyes in the presence of a stinger. And so in this way, if this model is favored, then there's evidence that traits are in fact correlated. However, the correlation could be a positive correlation or a negative correlation. And so to determine the specific nature of the correlation, I then conducted what I just kind of arbitrarily call a parameter restriction test, where I constrain pairs of traits that might be relevant to my hypothesis and test the fit of that model compared to the full dependent model. So the way this would be done is I would constrain, for example, these 2 dark blue arrows to be equal to each other. And then that would propose that the gain of large eyes is actually equal the transition rate regardless of the state of spines. And so if there's a significant reduction in likelihood from that model, then there's evidence that actually that difference really matters and is important. I think that this will be a little bit clearer when I show the results. I will in fact move to the results, and I'll start with just the phylogeny. Here you can see a phylogeny. However, even if you put your face really close to the screen, you wouldn't be able to see the tip labels, and that's fine. One kind of just takeaway from this image is just that there is a number of what these dots are showing is there was some results from the rate shift analysis, and you do see that there's a variety of rates of diversification and shifts in diversification in this tree, which would kind of beg an explanation for why this has happened, and it may have to do with the defensive traits. This phylogeny includes 268 ant genera, which at the time was 82% of all described ant genera. I earlier referred to 271. That's because I had to prune a few tips that didn't have sufficient trait data. I also found that the topology was consistent with earlier studies, as was the dating, and so this phylogeny was used for some subsequent analyses. I'm going to start with the correlation test, which again was targeted towards assessing evidence for a trade-off. This figure is a little bit overwhelming at first. What's shown here are the results only for the statistically significant pairwise comparisons done. I actually did a lot more than these. An example of a positive association that I detected is shown here. what you see is that there's a large, that the loss of spines when you have small eyes is more likely than the loss of spines when you have large eyes. So this would be kind of a sort of positive association where if you have both spines and large eyes, you're less likely to lose your spines. But then an example of a negative association is shown here between spines and a stinger. And what you see here is that the loss of a stinger is more common if you have spines than it is if you don't have spines. So this I interpret as an evolutionary trade-off, where something about having both spines and a stinger promotes the loss of the stinger. And in this way, you can see that there are 2 positive associations and 4 negative associations. All of the negative associations are between a stinger and the other 4 defensive traits I looked at. And the positive associations are between spines and large eye size, and large colony size and large eye size. And so I do in fact find evidence for an evolutionary trade-off between these kind of suites of traits on one side and the stinger on the other side. For the diversification analyses, what I'm showing here are the results from the sister clade analysis. The N down here is showing the number of sister clades that I was able to use for each trait. And so there's actually quite a large number of sister clades for each of the tests, which is good for the statistical properties. We find a positive association between diversification and several defensive traits, as well as a negative association for spines, for a stinger. And I do want to highlight that spines, again, come out as one of the important traits in ant evolution. So taking these results together for this chapter, one of my take-homes is that spines are a key trait in ant evolution, along with other defensive traits. And in particular, several defensive traits, including spines, seem to trade off with the stinger, and spines are associated with increased diversification in ants. So this both highlights the importance of defensive traits, which have been somewhat overlooked in ants, as well as highlights the importance of spines. And then even for the stinger, which I don't pursue in my further work, but this would be something that would be interesting to probe in the future, why a stinger trades off with traits, and it may have something to do with the complexity of its structure or the kind of active nature of its use, or several other potential mechanisms maybe driving the trade-off there. But so spines are really important. In ants, spines are actually quite common, and they're actually very variable. I didn't present this, but in my first chapter, I do find that there's over 100 genera that have some amount of spinescence, some amount of spininess. A previous study that's fairly recent and came out during my PhD found that using one polyvacus species and one tree frog in the genus Hyla, found that spines do increase survival against a vertebrate predation. The spine traits are also highly evolutionarily labile, which I found in my chapter, and was also found in another study. And there's been some support for links between the spine traits and various different habitat and morphology traits. For example, Sarnat and colleagues found that there was a positive association between longer spines and more specialized inland island habitats. So all of this taken together, in addition to the results from my first chapter, really promote spines as an interesting trait to pursue in trying to understand ant diversity and success. I focus on a special spiny species, Polyrhacus, which has 700 species and which is remarkably diverse. It's the 4th largest ant genus out of, again, over 300 ant genera. And as you can see here, there's a really remarkable variation in morphology among spine morphology, among other traits. And it ranges everywhere from no spines. These species don't really live up to the spiny ant genus very well, the name very well. These span all the way up to this really long morphology that's like a fish hook that can extend greater than the length of the mesosoma, this middle section here. And so this variation and kind of extremity of traits lends this genus a lot of potential for trying to understand how spines might be influencing evolution in ants, and why all ants don't have spines while some do. Also, the ecology of this group is really interesting, and several species nest arboreally in the trees, and in fact, this subset of species are what I focus on for this chapter. And they nest between leaves and can actually use their larvae, which produce silk, to glue together. It's almost like an Elmo glue gun. They carry the larva in their mouth and they use it to glue together the leaves and other different substrate in order to nest inside of leaf cavities, essentially. And also, just as kind of an interesting factoid, the first documented case of true swimming, was in ants, was documented in Polyrhacus sokolova, a member of this genus. And also, many people will eat these ants for their medicinal properties in primarily in South China and Southeast Asia. So this group is really interesting both for scientific reasons, for ecological interactions, and also even for humans. And finally, they also have a very wide distribution, spanning all the way from Western Africa through Melanesia. And so it's possible that spines may be promoting the success and the geographic success of this group as well. So for this chapter, in order to try to understand how spines might be promoting ecological success or potentially driving a trade-off that might explain why some species have spines and some don't, I ask a number of questions. The first is, do species with spines invest more time and effort foraging? Is there a relationship between spines and competitive ability? And do species with longer spines have higher survival under invertebrate predator-prey conditions, even though there was a previous study that found an association with vertebrate predators? So for this first question, we might expect that there's a positive association between spines and foraging, as the workers are freed from having to worry about predators, so they can be more conspicuous and forage more robustly and consistently and persistently than species without spines. We might also expect that one reason why some some species don't have spines is that there is a trade-off where spines might be really good for anti-predator ability, but there might be some way in which they reduce competitive ability. And so we would expect a potential negative association there. And then spines may serve as very broad defenses, not just against vertebrate predators. And that's the reason for the third question. To address these questions, I also want to highlight that there was a study that came out recently that highlights that that top-down processes like predator-prey interactions have been largely ignored in ants. And so this study really fits into that gap, or that's one of my goals, is to try to fill that gap. So to address my three questions, I collected a number of colonies, and again, I collected their arboreal colonies, so there's these different leaf structures. And this was with the assistance of a number of excellent field assistants, including Einar, who's shown here. And I maintained these colonies in the lab with a food substrate of 50% sugar, 50% water solution. And I was able to collect 11 species and conduct trials in this multi-species framework, which is good for separating the impacts of spines from any other species impacts that that may be going on. However, for several of the species, I was only able to collect a few colonies. And so I also focus on a two-species trials as well, which, while there's some limitations for two-species tests for trying to get adaptation, these two species are very similar in many important ways, including they have similar body size. They have very similar kind of gestalt, general appearance and color, especially within the context of this very variable genus. They nest similarly, they have similar diets. But one key difference is that the Polyrhacus flavicornis on the left does not have a spine on the propodium, whereas on the petiole there is a small pair of spines, whereas Polyrhacus lavigata has a medium pair of spines on the propodium and longer pairs of spines on the petiole. So this is still a good species pair for looking at potential differences mediated by the spines. The specific trials that I conducted, first also I collected jumping spiders as well in the species Silurus semiglaucus, which is a vicious killer of ants. And it is adapted to and specializes on consuming adult ants. And this is another reason for including the two species that I did for the two species trials, is that they also are smaller sized and thus well suited to this predator. So the three trials that I conducted are independent trials, and they're addressing the different questions that I asked at the outset. The first are resource discovery trials. And the way that this worked is I placed the colony container after starving them for 3 days to ensure interest in the prey. I placed the container in a bin filled with water to prevent escape, because these are arboreal ants and extremely good at escaping, even with like chemical solutions to try to prevent that. I then attached the colony to a food resource using chopsticks, which are a nice standardized wood that kind of mimics twigs, and attach them to a food resource. And importantly, these are not attached to another colony. So this is a test of how good they are at finding food and how much they invest in food acquisition. For the competition trials, I connect a opponent colony to one colony, so that this is an assessment of the potential impact of food resource gathering when in the presence of another colony. And then for the predator-prey trials, I placed one adult worker with a jumping spider in a container, and I also starved the spider for 5 days to ensure interest in the prey item. Just for a little bit more detail about these 3 trials, for the resource discovery trials, I conducted these by counting the number of ants that were in the food chamber for every 1 minute over the course of an hour, and compared the results for the GLM and generalized linear generalized linear models and generalized linear mixed models. And these were conducted for both the multi-species and 2-species trials. I also then did the same thing for competition, except as mentioned, they were 2 colonies were connected. So I counted every minute the number of workers from each colony that were in the food chamber and in the opponent's chamber, as they could actually cross both chopsticks. And I used GLM and GLMMs for the 2-species trials, but for the multi-species trials, I was able to actually use a method for ranking competitive ability that is actually used in football, human football, and it utilizes something called the Colley matrix. And this is a method that's well suited for ranking teams for which all possible head-to-head matches that happen are much, much fewer than the total possible number of head-to-head matches, which is the case for American football. And so this is also the case for my colony pairing as well. It would probably have taken like a year or more of running trials to do all possible pairings, and the colonies probably would have died by that time. So I'm assessing resource competition and resource discovery. And then for the predator-prey trials, which again I conducted just for the 2-species trials, I checked survival after 1 hour and after 24 hours and compared these with a chi-square test. So to move to the results, which displayed here are the results from the multi-species trials on the left and the 2-species trials on the right for resource acquisition. And I also have displayed here the phylogeny for this group, which actually comes from my 3rd chapter, which I'll get to, with the number of colonies that's present for each species in these trials. For the 2-species trials, I also have a second number, which is the number that was used in the 2-species trials. So the take home for this slide is that there is in fact a positive association between spine length, which is on the x-axis, or in the 2-species trials, this is the smaller spine species with the longer spine species. And there's a positive association between spine length and all of the different metrics that I looked at, which was discovery rate, how quickly workers move into the food resource container, worker presence, which is the number of time bins for which there's at least 1 worker present in the chamber, which is a metric of persistence, and then worker number, which is an overall investment metric, which is the average number of workers over the trial period. And so we can see on the y-axis that it increases in all cases. And also I will note that the y-axes here are not on the same scale, so you should compare them directly. And in all cases, except for the discovery rate for multi-species, these results were either marginally or strongly statistically significant. And so overall, this suggests that spines are beneficial not just for anti-predator ability, but also do result in improved foraging abilities as well. For the competitive competition results, this is a phylogenetic regression, so I use the phylogeny here, where I correlated spine length against the Colley matrix ranking. And the ranking is somewhat arbitrary, but it was produced using the average number of workers from each colony, accounting for colony size, and used to rank the abilities of each species. But what we find is basically a flat line, and the p-value is almost 1. So there's really no evidence in the multi-species context with at least these 11 species that there is any association between spines and competitive ability. However, for the 2-species results, there's some slight evidence that there might be a positive association where the species with longer spines outperforms, it increases in its ability from its resource discovery trials. So this is when the colonies were solo versus when they were paired with a competitor. And you see this increase actually in ability when faced with competition. However, we also do see a little bit of an increase with the species with shorter spines. And so overall, this is kind of neutral evidence. There's not really strong evidence for a positive association between spines and competition. But if anything, though, there's some evidence for a positive association, but there's certainly no evidence for a negative association or any kind of trade-off. And then lastly, I do find a positive association between having longer spines and exhibiting increased survival when with the jumping spider Siler semiglarchus. This was not statistically significant after 1 hour, but survival after 24 hours was marginally significant via a chi-squared test. So overall for this chapter, we find that longer spines are associated with enhanced foraging ability. Potentially, there's some evidence that they might be associated with enhanced competitive ability, but certainly no evidence for a trade-off, and that they're associated with enhanced anti-predator ability. So taken together with my first chapter, so far it really seems like spines are really important for ants, both in terms of diversification at a broad genus level, and also at an ecological scale. It seems to improve their foraging abilities. And so if you recall from the first chapter, when I talked about the studies that find traits associated with diversification, some mechanisms that are proposed for this result are things like the escape and radiate hypothesis or the enemy release hypothesis, where evolving certain defensive traits frees you from the negative selection pressures from predators and allows you to radiate into novel niches. And in the case of the escape and radiate hypothesis, this may also drive some cycles of diversification as predators evolve to be able to predate on the group of interest once again. And so it's possible that this sort of dynamic is what's going on in Polyrhacus. And this is further supported a priori by the fact that they have this wide geographic distribution. It's possible that spines are one of the driving reasons why they are, Polyrhacus has been able to exhibit such a wide global distribution, or near global distribution. So in order to probe whether or not this is actually the case, I, of course, need to conduct some phylogenetic analyses and gather morphological and geographic traits to address these questions. And my core questions are spines associated with diversification in Polyrhacus? And is there a relationship between spines and geographic range, as you might expect via hypotheses like the escape and radiate hypothesis? Also, I want to note that there is an existing phylogeny from 2016 for Polyrhacus that included 80 species and was informative, as it included all but one of the Polyrhacus subgenera. But this is a group of 700 species, and so I want to significantly increase sampling to capture more species and also all the different morphological types that are found in this group. Furthermore, this phylogeny was inferred using gene markers, using 9 gene markers, and some newer methods are able to incorporate a lot more phylogenetic information by using genome-wide approaches. And the specific genome-wide approach that I used was something called ultra-conserved elements. And what these are conserved regions across the genome that are conserved across ants and oftentimes across Hymenoptera and even other animal groups. And you select, in the lab, select these conserved regions, but also sequence the flanking regions, which are more variable. And in this way, you can get homologous genetic information that you can compare across groups, but that also includes enough variation that you're able to resolve recent divergences as well as more distant divergences as well. So in order to conduct this approach, I followed a standard pipeline by extracting DNA and targeting UCE loci. And for this step, I was able to use some existing DNA that was already extracted from the previous Polyrhacus phylogeny study that I cited earlier, Metzger and Moreau. And then I followed a standard phylogeny pipeline and conducted phylogenetic inference in a variety of different programs. To say a few details about the inference, here's this really boring block of text. The analyses included 160 Polyrhacus taxa, and I targeted 2,524 UCE loci, as is standard in the ant-specific UCE bait set. That also includes Hymenoptera UCEs as well. And I resulted in 1,300 loci in the final data set, and this was because I used a 70% taxon complete matrix, which is an effort to try to maximize overall data usage while also trying to have fairly good quality data. And what this means is that for each loci, there's some data for at least 70% of the taxa included. Then I conducted maximum likelihood inference in RAXML, Bayesian inference in Exabayes, and gene tree approaches in Astral3, and then conducted divergence dating in the program MCMC-tree. And for the RAXML analyses, I used an unpartitioned, gene partition, and R cluster F partition data set, the latter of which was in partition finder 2, to try to determine what the best segmentation of the data was. And in this favored R cluster F, which was used for the Exabayes analysis as well. I conducted bootstrapping using the auto MRE function, which determines bootstrap number automatically. And I used the GTR plus gamma substitution model in all analyses for which it was relevant. and conducted divergence dating again in MCMC tree. And unfortunately, I don't have informative fossils to use. So instead, I used uniform node constraints, which were from previous studies, the previous Polyrhacus study that I cited, a previous phylogenomic study of Formicidae, which is the subfamily that Polyrhacus is a part of, and a genome-wide phylogenetic analysis from 2013, Moreau and Bell, which I use these three studies to constrain or to put a prior on the crown age of Polyrhacus, as well as some of the outgroup taxa as well, which were used from the phylogenomic study by Blamer and colleagues. In order to use this phylogeny, of course, I want to also have traits to get the questions that I'm interested in. So I gathered morphological data, and this was a fairly straightforward approach. What I did was I gathered images from AntWiki and AntWeb, and in the program ImageJ did linear measurements in order to capture body size and spine size. In order to measure spine size most accurately, I used the view that was most parallel to the plane of projection of the spine. I was able to get images for 154 species, and about 82% of these have at least 2 images or more, and I used a maximum of 3 images for each species. For the species that had 2 or 3 images, I averaged the measurement totals for my data in order to account for natural variation and also potential measurement error as well. For the geographic range size, I gathered this data from antmaps.org, and I used the political designations because what this resource provides is the distribution of species and all of the political categories, political geographic designations where they occur. So even though this isn't quite as precise as the more natural boundaries, this is still captures quite a bit of variation and is still better than using really broad categories like the Indo-Pacific region. And my data set does span from 1 to 51 territories. So this is showing that there is a decent amount of variation, inter-specific variation that's captured in this approach. So now I will move to the results. First, what I'm showing here are the dated phylogeny, and I'll walk through what the colors and bars mean. This phylogeny, MCMC tree inferred a crown age of 33 million years ago, which is fairly similar. It's a bit younger, but it's fairly similar to the previous Polyrhacus phylogeny. The colors are showing the evolution of spines along the tree, and what we see in the warmer colors, the reds and yellows, are smaller spines, and the cooler colors, like the greens and blues, are longer spines. And so you can see there's a number of places along the tree where longer spines or shorter spines have been evolved again or independently. And so this suggests a decent lability in the increase and decrease of spine length in this group. The bars are showing geographic range size, and these, you can see here, is also quite a bit of variation and convergent evolution of geographic range size in this group. And somewhat interestingly, although I haven't probed it too much yet, there is one group that seems to uniformly have fairly small geographic range size, but the bulk has a decent variation. In order to understand, if the geographic range size and spine size are correlated at all, this is one of my questions. I of course want to conduct actual analyses, not just look at it, because you could squint at it and see patterns if you wanted to. But also I conducted trait-based diversification analyses as well, which I will talk about first. The trait-based diversification analyses, for these, I actually found that spine size was not correlated with diversification, which is in contrast with my first study, the results of our first study. And really, this was also consistent across all 3 different analyses. What's shown in the pictures here are the results from BAM, but I also conducted analyses in these 2 different methods, QASI, which is a state speciation and extinction method, and ESSIM. And when you look at these plots, you might see that there's this one burst here, or it looks like a burst of increased diversification. But this is actually not statistically supported. In the BAM analysis, if you look at the 95% credible shift set, which is the set of shifts for which at least 95% of the data is explained in the analysis, we can see with these frequencies here, which is the percentage of the time that this shift was favored in the simulations. You see in the inferences, you see that there were actually 52% of the time there's not the shift found, this burst in Sirta Mirwa, although in 42%, or roughly 40% of the time there is. And also, even if this does hold to be true, this is actually the genus Polyrhacus pseudomyrma, which is spineless, not because it's isn't courageous, but because it doesn't have spines. And so even if this is actually proves to be true, there's, if anything, a negative association between diversity and spines and diversification, according to this analysis. So now taking all three of these studies together, all three of my papers, I'm jumping ahead of myself. I'm getting so excited. I want to get to the conclusion. I also, of course, want to ask about the association between spine traits and geographic range size. And what we find here on the x-axis here is spine size, and y-axis is range size for the and then for this plot is body size and range size. For these two, we see that spine size actually is associated. It has a positive relationship between range size, even with all this variation, which supports the hypothesis that spines are promoting geographic range expansion in Polyrhacus. And I did not find any association between body size and range size, even though this tends to be important in other animal groups. Also, I did find a linear relationship between spine size and body size, which you might expect as species get larger, they will just have larger traits in general. However, I did not find any allometric scaling where there's no disproportionate increase or decrease in spine length as body size gets larger. So now, taking all these results from these two chapters together, I find pretty consistent evidence that spines promote ecological success. And this was true both for foraging ability, anti-predator ability with an invertebrate predator, and geographic range size. Whereas I find some more mixed results for diversification, where with the genus level phylogeny, which included 82% of all genera, I do find a positive association between spines and diversification. Whereas for the Polyrhacus study, which I will note includes only 22% of the genus, I do not find an association. But it is important to remember that the sampling for this phylogeny, although it is a species level phylogeny, which contrasts with the first study, I would really be interested in increasing sampling to try to get more to determine if this relationship actually is true. And I'll get to that in a moment a little bit more. So overall, another kind of general take home is that defensive traits really do warrant further study in evolution and ecology. I cited the earlier study that highlights that top-down processes like predator-prey interactions have been woefully understudied in ants. And I think that when we're confronted with all this variation in traits that have at least a partially defensive function, it would really behoove us to probe the role of defensive traits more, both for the ecological success of ants and for the evolutionary success of ants as well. There's a number of kind of future directions that I might go with this work. As I mentioned, I would really like to increase sampling. And I would like to get to at least 50% of the genus, but I might as well dream big and shoot for 100% of the genus, or maybe 80% or 90%. I also have, during the course of my PhD, conducted a number of, I conducted scanning, micro-CT scanning of the shapes of samples of Polyrhacus and did some preliminary 3D morphometric method analyses on the preliminary data set. And this method really, which I would like to thank the Low Lab and April Neander for assistance with this part. The scanning of these in this method allows you to ask really interesting questions or more detailed questions, like the potential association between spine length and cuticle thickness, for example. And there's a number of other questions you could ask. So I'd be interested in building off of the morphological analyses that I've conducted for this dissertation. I would also like to conduct more experiments actually within the field. The trials that I conducted for my second chapter, while informative, did occur in the lab. And so it's possible that the behavior shifted in the lab, and there's a lot of other dynamics that could be going on that aren't incorporated with those lab studies. So I'd be interested in potentially using canopy cranes, which are, this one is from XTBG, which is my field site, Xishuangbanao Tropical Botanical Garden, and using this canopy crane and others to understand the interactions between Polyrhacus species in their natural environment. And there's really so much more, from developmental work to genome-wide association studies, where you could try to understand the gene networks that are actually driving spine gains and losses. And so I hope to continue working on this interesting group in the future. So that's the bulk of my talk. But of course, in order to complete this work, there's a number of people that I want to thank. and also even just for my development as a person, which is really important to doing anything. I really want to have a number of people to thank. So I will do the acknowledgement section, and after this point, as Corey mentioned, we'll do the Q&A where you can ask questions that you want, any questions you might have. I also, because this talk is being recorded, I felt a little bit weird about including pictures that people might not necessarily want recorded. So I found a way around that. And I would like to start with thanking my parents, whose names are Mama and Daddy. And of course, obviously my physical form wouldn't be here without them. And I also am just really appreciative for your the kind of my intellectual development, spiritual development, emotional development that you promoted from a really young age, and in particular even for ants. I actually while I was preparing for this dissertation, I pulled out the book that you gifted me, which is Amazing Insects, and I wasn't sure if this was the right book actually. This was gifted to me between my junior, or for my birthday, I think, of my senior year of high school. But then I checked the date, and this is in fact the book that it was published in 2008, which checks out. And I flipped to the page about ants milking aphids, and I was pretty blown away, because this is in fact what I always cite as the reason why I got interested in ants whenever anyone asks me, which has now happened over 100 times. And like the wording of this is almost identical to what I say all the time when I'm sharing about ants at the Field Museum, or if anyone asks. And so I'm going to read it aloud, as it was very informative for me. Milking aphids. Ants love sweet things, and one of their favorite foods is the honeydew exuded by aphids. Here, an ant is stroking aphids to encourage them to give out the sugary liquid. This is known as milking, and the aphids are frequently referred to as ant cows. In return, the ants protect the aphids from enemies and sometimes tend the aphids in their nests. Aphids feed entirely on plant sap, which is rich in sugar but relatively poor in protein, so to obtain enough body-building proteins, they take in far more sugar than they need. Most of it goes straight through and emerges at the other side as the sticky droplets of honeydew that the ants love. So in case you missed it, that means that they're pooping out pure sugar, essentially, that the ants are eating. And then it says, ants are so keen on the honeydew that they get from aphids that they often carry them to fresh pastures. Here, the common black ant, Lasius niger, is gently carrying a young aphid in its jaws. It will deposit the aphid on a fresh plant where it will find good quality food. Some ants even build cowsheds around their aphid herding by piling soil particles around the bases of the plants, and aphids are also herded in many ants' nests, where they feed on roots and underground stems. So, I mean, it's pretty amazing stuff, and as far as I'm aware, that is the reason why I got interested in ants, which I then continued at the University of Michigan. So, obviously I could go on, but I'll stop there, and also thank you for the food and shelter too. That was pretty nice. Next, I'll move to my brothers, which is Joshua, Evan, and Jordan. Joshua is a philosopher, and Evan is a music teacher, and Jordan is a music therapist. So two of you have useful jobs. And I really am so thankful that I have brothers that sense sources of comfort and joy and friends. You really are my first friends. And I also would like to especially thank Josh for nothing, because had it's pretty remarkable that I actually finished this PhD given all of our Google chats. I think our Google chats could probably span the length of 2,000 dissertations. So I really want to thank my own persistence for getting through those and still being able to finish my dissertation. Next, I want to start with my family, but I can also start with move to my Ant Lab family, and I really want to thank you, Corey, for just a really remarkable 5.5 years. Whenever people ask me about whether or not they should come, and if they think I should go work with Corey, or if they ask information about you, I always say that I would if I redid my PhD, I would still join your lab in a heartbeat, and that's just so true. What I really appreciate is the way that you treat your students, including me, as I didn't think I was going to cry. Now I am. As whole people that you're not just invested in our the research ability, but or trying to extract data from us or information from us. You really are invested in our development and supporting who we are and what we're interested in, and that's something that's been quite consistent throughout these years. And so I'm really I'm just really so grateful for your guidance and for making and supporting the Ant Lab as a second home for me in Chicago. Next, I would like to thank my committee, which this is Kathy, Graham, and Trevor. I often tell people I have a shockingly functional committee, which is a little bit of an indictment on general academic structures. But I really thank you so much for your feedback. It's been consistently helpful and reasonable, and I look forward to getting more feedback at the end of this talk. I'd also, of course, like to thank my fellow YAS Queens, the Young Ant Society Queens, Jordan, Peter, and Supriya. This is based, in very stunning detail on a picture from Facebook, and this is showing Jordan getting his second master's in science and Supriya getting her PhD degree. And I mean, there's like just so much I could say for you four, but I have for you three, for us four as a group. I just thank you so much for all the kind of wild, sad, happy, joyous, absurd times that we've had internationally, whether that's in Vancouver, or in on the East Coast, or in Boys Town in Chicago, and I really look forward. I'm actually almost like less sad in some ways in thinking about you three, because I know and I'm very confident that we'll stay in touch and see each other so often. Even you, Jordan. I know you're going to try to disconnect, but don't do it. Shauna, I hope you're listening in. I know you also potentially had teaching duties, but I feel like I probably wouldn't have finished my PhD without your both emotional and academic support. I really, it's always hilarious how similar a lot of our research interests are, and I especially really thank you for being a fellow snack lover. I maybe I would have withered away without your assistance, and I'm really looking forward to work with you in a short-term postdoc this summer, and along with Corey, of course, and I'm happy that you're still based in here in Chicago. So we definitely need to talk soon. Manu, again, there's too much I could say. One thing I'll say is, você sabe que as beads não são a verdade, né? And probably some other Portuguese speakers won't necessarily understand what that means, but I know you do. I am just so blown away by your energy and positivity, and I really, even if I always think of things are going to fall apart and go wrong, you always seem to somehow maintain this positivity while also still being really passionate and committed about things like social justice and other issues. And so I really just thank you a lot, and I can't wait to talk with you again soon. And then just the whole Moreau lab, I really, the Moreau Ant Lab, I really, there's so many to thank. I've just put a few here. If you're not on here, it's partially because I did have to stop drawing at some point. It's not anything against you. Here you can see Max and Ben holding their taxa of interest. They were former grad students. I really thank you for welcoming me into the Ant Lab. Matt Boot, I really thank you for your, he's holding a Polyrhacus specimen here. If you look closely, it's actually on the pin. And he assisted me quite a bit with preparing the specimens. I really thank you a lot. Destiny, this is a very bad rendition of your hat, I'm sorry, holding a 96 well plate. I would not have completed the UC analyses without your help. Andrea, I really enjoyed, you were a member of the lab right when I started. We're sharing an office, and I've enjoyed being part of the book club with you as well. And of course, thank you to Emily, Matt, holding his lichen, and Anais and Christoph. And I put the chemical equation for formic acid there just for you. I now get to Natalia. I know I keep saying that there's so much I could say. For Natalia, I also would not have finished my PhD without you. I'm not sure I would even still be here without you in some ways. You really just enhanced my development as a person. You getting to know you and on all of your awesomeness has just been remarkable, and you're a true inspiration to me. I really hope that the world kind of catches up to your beautiful view of life, where I think you really see people as people, and even in an institution, you always are committed to trying to improve the way that wherever you are in your space. And I really just thank you a lot for showing me that. And I think you've improved me as a person and just been a great friend besides. And so thank you. I also want to thank CB et al., which is the Ecology and Evolutionary Biology Department or Integrative Biology, and Integrative Biology Department or Organismal Biology historically. And I have some people here. I'm sure you all know exactly who you are just from this really good rendition. But this is Laura, Christina, Jackie. I actually decided to draw I think it's a humorous from your recent publication in PNAS. Hopefully I did a good job. I also enjoyed to play tennis with you, Andrew, even though it's like then we kind of stopped playing, but I think we only played like twice, but still it was, I still cherish those memories fondly. Heather, I thank you for all the many chats that we've had over the years. And Mariah, I always enjoyed having dumplings with you. And there's just so many other people to thank, which again I just couldn't put on these slides, but I really thank all of you for creating a wonderful community in our cluster of departments. I also want to thank Carolyn Johnson, who was our administrator for many years, and it remains an injustice that you were let go due to corporate restructuring of the university. I thank you very much for all the goldfish, and I always liked your myriapods as well. I hope you're doing well. I'd also, of course, need to thank my colleagues at XTBG, the Xishuangbanna Tropical Botanical Garden in Yunnan Province. This is Dr. Cao and Dr. Akihiro Nakamura, or Aki, and I really thank you for hosting me in your vibrant lab. And there's, of course, so many others to think of the lab: Song Xiaoyang, Johan, all the administrators like Yang Xiao, Xian Hui, although I know you're doing grad school now. And I really thank you for all your assistance. Of course, this project wouldn't have happened. Both samples from my last chapter and, of course, the trials in my second chapter all come from being able to come do this work and collaborate with you. And last but not least, I must thank my 4 wonderful field assistants over a period of 3 years: Chenny, Linarong, Renming, and Einor. I thank you for suffering through a few ant bites, but at least the ants can't sting, so we kind of lucked out there. I also want to thank the Field Museum, both in terms of its institutional support and all the administrative support, but also the education department, which helps to facilitate and promote science communication. And I really enjoyed participating in those programs. And I thank you for conducting all that work, which I know can be more difficult than it looks. And of course, I need to thank the Pritzker lab as well, the Pritzker DNA lab, Kevin and everyone else who keeps that lab running. And I thank you all for being awesome people. And then I need to thank my funding sources as well. These include the Xishuangbanna Tropical Botanical Garden, NSF, as Corey mentioned, I got a few NSF grants, which they graciously gave me. And then also the Field Museum, I had a fellowship for 1 year there, and various funding sources from the University of Chicago, including the Committee on Evolutionary Biology in my department. So that was kind of the academic thanks. I also have a number of other people to thank that have kept me afloat and as I've mentioned, helped me become the person I am, and are continuing to make me better. I would like to start with Natalie, and what I'll just say here is thank you for letting me be the dragon. I also want to thank Nathan, Andrew, and Ryan, and this is Anna, who's running off somewhere probably. And I really thank you for just being such a great group of friends that I can just completely be myself with. It's really wonderful. This is true for many people. I did come out as gay during grad school, and I really it means a lot that that I'm able to even friends that I've been friends with and not out to for almost a decade, that I can still feel comfortable chatting with you. And I also of course have enjoyed our D&D group, and it's been great getting to know you, Samantha, as well. And I also thank you all for mourning the sad loss of my dwarf sister, Bjorta. Jordan, Stephanie, and Claire, you guys are such a great group of friends, and I got this picture of us 4 here together, and I really can't wait to see what kind of adventures we do next. Katerina, I had this WhatsApp symbol here because I think without WhatsApp, I would be a different person, but I really love all of the the chats that we have, chatting almost every day, and becoming closer friends with you has been a true joy these 5.5 years. And thank you so much for the book that you sent to me, and also the donuts at my proposal defense as well. It really meant a lot to have that support from afar. Carlos, with a C, you start doing karaoke every Wednesday night has really has not been something I ever thought I would necessarily do. It required having to go to bed a little bit later some nights. But I did promise you that I would sing a song with you as for celebrating the defense. So I'm going to sing a little bit of a song, which was the first song that I ever sang with karaoke. And I also sang it actually with friends and colleagues at the Shishabana Tropical Botanical Garden as well. So it kind of connects multiple different worlds. And you can feel free to sing along with me. Can you feel the love tonight, tonight? It is where we are. It's enough for this wide-eyed wanderer that we got this far. And can you feel the love tonight? How it's laid to rest. It's enough to make kings and vagabonds believe the very best. It's enough to make kings and vagabonds believe the very best. Well, got that down. And I have to thank Aileen for just, I think the best way to put this is just thank you for being all that is sparkling you. I also would be remiss to not mention the Daily Ant, and in particular, one of the series that the Daily Ant has hosted in the past, now I think it's over 2 years of its existence, is the Philosophy Fridays series. And I need to thank Catherine and Lauren for being the best executive producers of the Philosophy Fridays series that a guy could ask for. And I also want to thank all the different philosophers, I think some of whom have tuned in, who contributed to this series. And it was really fun to be able to, have an outlet for my interests in, kind of sharing about ants and communicating to different spheres. And I never thought that one of those would be philosophers and creating a series that joins ants and philosophy together. But it's really been a true joy. And if you want to follow the Daily Ant, you can do that on Facebook and also on Twitter as well, or sign up to the site via email. Yes, that was a plug. And of course, I need to also thank Julia Debress, who's the sister of Helena Debress, who is one of the philosopher contributors to the Daily Ant. I also really want to thank my church, Ellis Avenue Church, the little house on the corner. I'm always thankful to Pastor Jonathan and Pastor Georgia for your wonderful spirit and all of you. In my actual document dissertation, I list many names of everyone at the church. I entered this community at a time of really significant grief for all of you, and that became my grief as well. And it was really beautiful to see how you mourn and remember the loss of members of your community. And it showed me something about the church that has held true to this day. And one of the main things also has kept me is your commitment to social justice. And I really think you all just show what it really means, what it should mean to be followers of Jesus. And I really thank you all for that. I also need to, of course, thank my wonderful housemates who have become especially housemates during this situation we're all in now. I need to thank Bob. Apologies for the as gray as made your hair. My abilities are not great. Hibiki, and then it's also been great getting to know you more, Amy and Puppy as well. I'm really not ready to leave, Bob, so if I could, it'd be nice if I could just stay indefinitely in your apartment. Thank you. And lastly, but definitely not leastly, I would like to also thank everyone who is active members, even just supporters of Graduate Students United, our graduate worker union here on campus. As many of you know, we conducted a National Labor Relations Act monitored election, that high turnout and we won that election, our unionizing election, by a supermajority margin of about 70%. And still, the administration, despite promoting us to go vote, didn't accept that effort, and has been stonewalling and being oppositional since. And so some of my kind of feelings when I think about GSU, which I was involved as an organizer for quite a while, is some of it's kind of anger at the administrative response, and kind of opposition even among many faculty in BSD. But overall, I'm really just filled with like such gratitude and kind of amazement at all my fellow grad colleagues in BSD and elsewhere, as well as community support, both within some sections of the university and more broadly in the community. And one of the hardest choices for me was actually pulling back from union work this past year as I'm finishing my dissertation. And I'm in deep awe of Natalia and many other organizers who are able to kind of juggle things a little bit more. And one of my kind of key goals for wherever I land next is how to balance kind of working and paid work with other sorts of social justice and activism that's important to me as well. So thank you everyone at GSU, and also I also want to thank all of you as well. Obviously, there's just a ton of people I didn't mention, including everyone in the special WeChat group, which you know who you are, from XTBG. Just a whole number of people, but you all mean a lot to me, and it really does take a village to study ants, I guess. And with that, I will take any questions. And
Great,
I guess I will.
Thank you so much, Benjamin. I just want to take a moment and say at this time we'll transition to the public question period of the thesis defense. So if you would like to ask a question, please use the raise your hand function in the participants button on the bottom. But also please remember to unraise your hand once we have unmuted you to ask your question. Also, I hope everyone will join us on the other Zoom link that I pasted in the chat window, which I will do again at the end of this question period. In 1 hour, pending successful meeting with Benjamin and his thesis committee to congratulate Benjamin. So with that, we'll be taking questions.
Yes, let me also try to get this so I can get we open the chat. I'd like minimize things in order to, I was doing that. Sorry, I am, oh, it popped out as a pop-out, sorry. So I'm just getting used to this system.
So we did have a question from someone who was here, but had to leave for another Zoom call. And the question is, defensive traits in ants influence individual survival odds and productivity in workers and soldiers. But since they are not reproductive individuals, they only cascade into the fitness value as far as selection is concerned, though through the overall productivity of colonies. Is there a way to link or predict the impact of individual fitness conferred by defensive traits in workers and soldiers on the overall fitness of the colony?
Yeah, that's a really good question. Yet again, that would actually be one of the things I would actually like to expand upon in my research if I continue doing this. One of the ways that you could link it is by trying to track survival or in particular the reproductive output, the number of larvae and kind of success to adulthood of larvae in the colonies, and could potentially associate things like spine traits with that. It is a little difficult because, of course, in order to like get at the colony numbers and like the number of workers and number of larvae, you have to you have to like destroy the nest already. You can't count them before they're done. So there is some complexities to that, but absolutely, I agree that ultimately, if you really want to super robustly establish fitness advantages, you would want to look at the reproductive output of the next generation. So that's a great question. And
people are welcome to post questions in the chat window too if they.
Yes, for some reason I'm having.
But we have some people who have raised their hand. So Tristan, we're going to unmute you so that you can ask your question.
Perfect, thank you. Hey Ben, I'm kind of wandering myrmecologist. Hannah Marty passed the link to your defense on to me. I hope you don't mind. I listened in. What a spectacular presentation that was. Very exciting. I work on army ant warfare with other ants, and so these defensive traits are obviously of great interest to someone who's looking at that. So I was curious if Polyrhacus has any sort of collective social predators in any of those species.
Oh, like social predators. So you mean like if like army ants will predate on them, or like that sort of?
Absolutely.
Yeah, it's a good question. I don't actually know for sure, to be honest. Actually, that would be an interesting thing to look at. Yeah, I mean, because yeah. So basic answer is I don't know. I'm not sure. And I'm actually gonna add that to my list though of things to look into, because I've often kind of thought of other ants more as in the context of them being other competitors, and then other non-ant species as potential predators. But it would be interesting because there certainly are some army ants that forage in my field site, for example. However, I also did focus more on the arboreal species, so they're probably not predating on those ones as much, although some of those do also go down to the ground level to forage. So yeah, so like that would actually be interesting to see if the spine traits at all might impact the competition or the resistance from social predation as well.
Spectacular, yeah. Thank you. Great presentation, and best of luck in your next round of your defense.
Thank you so much, and thanks for the question. And I saw that there was someone else who had their hand up, although I think they're gone now. You might have to put the hand up again if you did the hand.
Any other questions from the general viewership? Again, please
yes,
feel free to put them in the chat.
There's Nadia. I've unmuted you, Nadia. Welcome.
Hey, Ben, great presentation. I'm not sure if I missed something or if, perhaps you maybe didn't go into this as much, but I was wondering why having spines might increase foraging ability.
Yes, actually, thank you for asking this question, because I had meant to talk about some more detail. I think I maybe mentioned it in like a passing sentence. But yeah, so my thinking on this, I mean, it's somewhat speculative, but is that by evolving spines, the workers no longer have to worry about being predated on, which, especially by vertebrate predators, would be promoted if they're super conspicuous and really obvious in their environment. And that by evolving spines, they're able to therefore invest more workers without being worried about like a significant being wiped out by some big predator like a pangolin or something. No, that'd be more colonial, but or also and also being able to be more kind of just like consistently persistent. They can spend more time at the food resource in order to without having to be worried about being attacked. And so those are some of the potential reasons why there might be an association according to my thinking. I don't know if that makes sense to you or not, but all right, I think you've remuted yourself.
So we have
Thanks for the question.
A question in the chats from Catherine. What is your favorite thing about ants?
What is my? I think she asked me this because she knows I during some time I would ask other people, and somebody try to get some like famous people and stuff to try to answer that question. But yeah, my favorite thing about ants, it's very hard because there's so many things. But I do think it's kind of like actually what I referred to with the book that my parents gave me, which is the kind of like intricate nature of their interactions is just really stunning. Like how wild is it that these ants can carry an aphid that like sucks sugar out of a plant and can like tend it and literally milk it like a cow and like move it to greener pastures, like pen it in, protect it from predators. It's just really wild, and that's just one species too out of the 13,000 species. And so I think just the kind of behavioral complexities that are similar to humans. It's kind of one of the reasons why lots of lay people who don't study ants enjoy ants. Behaviors are just really pretty fascinating to me. And then I have gotten drawn into also the remarkable physical variation as well that's kind of associated with that. So that is what I would say. Thank you for the question, Catherine.
Let's try to maybe keep the questions related to his research, at least for this part of the thesis, instead of just general questions, which you guys can all ask him during his congratulatory Zoom call. Have a hand raised?
Yes. Hello.
Hello.
Hey, Parnell, how are you?
Good, how are you? Great presentation.
Thank you. Great to hear.
I was just gonna ask, I'm kind of curious what, for your simulations, what software tools or like how you did your simulations, like if you had software tools that you used. And
Oh
then I was
yes. Oh,
also,
sorry.
yeah, and then I was also gonna ask, I didn't quite understand about body size and spine size in terms of their correlation and like it seems like spine size makes them able to distribute further into further geographic regions, but not body size. But it seems like the two are also correlated. So I just wanted to understand that better.
Yeah, so those are good questions. For the first one, for the programs, it's really kind of a variety of different programs and different software. Like sometimes the software is created by the developers of specific methods, but also oftentimes I would use things like R or implement it in RStudio. Some of it's also just like in terminal using Unix. Some of the programs are Python based, and there's just kind of a number of some of those are like packages also, so I don't have to like do all the coding for that. But yeah, so it's kind of a number of different softwares. And then for the correlation for bias size, one of the reasons why I kind of included bias size is kind of as an alternative to spine size. Is just a trait that is really important in a lot of organisms, a lot of species, and then also it's also possible that because spines like these ants in this genus vary in their overall body size from quite small to actually pretty decently sized. For ants, and so probably like an order of magnitude in terms of like length. If I had to guess the back of my head, maybe like 5-6 times variation in size, maybe even 10 times. And so if I just did spine length, then it would have it potentially is just capturing body size. And then I also want to correlate body size to spine size, because it's also possible there could be some situation where similar to actually my reply to I think it was Nadia, the increasing body size may also make you more conspicuous to predators, and so it's possible that the pressures to have spines could actually increase as you get larger, so there'd be disproportionately high amounts of pressure with larger body size. And if that was the case, you would expect an allometric relationship, which I did not find, which would be a disproportionate increase in spine length as you get bigger in body size. But I did not find that. So those are the kind of reasons why I looked at what I and then the reason why body size would spine size could be correlated with body size wasn't with geographic range could also be that even though I do find a positive relationship between body size and spine, it's not exactly 1-to-1, and so it's so that could be an explanatory factor there.
Great. So we have a question from Mike Coates. Mike, I'm going to unmute you.
Oh, hi, Ben.
Hey, Mike.
Great talk. Thoroughly enjoyed it.
Thank you.
Naturally, I'm interested in the spineless ones. You showed a tree early on with the branch color was correlated with spine length, and you discussed one clade, but it gave suggestion that maybe that spine reduction had occurred 3 or so times. Have you got any thoughts, speculations on what's going on in any of those clades about spine reduction? Any correlations there you think might be linked to performance?
Yeah, so I do think that one possibility is that in the group where I found that one potential burst of diversification, the Cirtumira subgenus, they have also evolved this behavior where they actually will drop from trees or from the leaf. Like if you just touch the leaf, they'll just hop off the leaf and just fall. So it's very possible that they basically evolved they've independently evolved this behavioral defense to possibly even compensate for not having spines. And so that's a potential explanation. Also, even though I don't find yet any negative, any trade-offs between having spines and the other various different things I've looked at, I still kind of, one kind of has to assume that there's at least some trade-off somewhere, otherwise all ants would have spines, if it's purely beneficial. And so I'd be interested to look at maybe those developmental trade-offs. Potentially there could be spatial trade-offs where it limits what kind of nest spaces you could occupy. So potentially the species with smaller spines are able to, at least within microhabitats, invade more space, like cavities and stuff. So yeah, those would be some things I would be interested in pursuing. But yeah, I was a little bit surprised actually to find like so little evidence of negative associations between spines and the traits I looked at.
Thank you.
Great. We have a question
Yeah.
from Shannon Hackett. So Shannon, I will unmute you.
Hey, Ben.
Hey, Shannon.
I have a question about kind of robustness of spines. So if spines can be used obviously for defense, as you're saying, but as an ornithologist, I have to ask you if they can also be used for display behaviors. And so have you thought about how to measure not just the spine's presence or its length, but kind of other aspects that may influence its function?
Yeah. So I'll do like two-part answer. The first is that, and I mentioned this in the publication that's it's in work for this in progress for this, but that these spines, yeah, like are almost certainly not sexually selected, because they're significantly reduced or absent in the reproductives, which is the queens and the males. Males don't have, I don't think there's any Polyericus species for which males have spines, and the queens in some cases have, like for the species where the workers have really long spines, the queens might have some like smaller spines, but it's very unlikely that spines are used for display. But even still, like ways to capture more information about the shape as opposed to just linear length would be things like potentially scanning, like I conducted with micro CT scanning, and I would be interested in trying to get more at like the shape, because not only may there be some other functions I wasn't aware of, but even for the anti-predator function, it's possible that, like, for example, curved spines might be more well adapted to vertebrate predators, or like large vertebrate predators, or maybe like those large invertebrate predators that the more straight spines may be well adapted to as well. Yeah, so like those are some potential things that someone could do, I would say.
Thanks.
Yep.
Great. So we have a question from Haimander Bharti, and he asked, is there a positive correlation between proportional spine length and species richness?
Proportional. Yeah. So actually, so sorry. You don't know if you can put in the chat. Do you mean like without the phylogenetic information, like it's just purely species richness and spine length?
Maybe you could answer it both ways. And
yeah, because that would be best. Yes. So I haven't done. Actually, I didn't correlate it without incorporating phylogenetic information, but with phylogenetic information, at least within Polyericus, the answer so far seems to be no. That there's, because the spine length metric, I actually think I failed to mention this. The spine length metric I used is the relative spine length. It's a spine length divided by body size, and there was no association between diversification. I actually didn't do it with just richness, like maybe like a PGLS or something with just richness. I haven't done that yet. I guess I could do that just to see what it might show.
Great. So we see Mariah Scott has her hand raised.
I can unmute.
Hi.
Hey, Mariah.
Hello, Ben. In your chapter 1 discussion, you had several significant relationships, and many of them seem to be trade-offs between traits, but I remember there were 2 that were not seen by you as trade-offs, and I was wondering if you could speak to any thoughts or hypotheses you had about that.
Yeah, so those positive associations were between large, where spines and large eye size, and large eyes and large colony size. And one of my thoughts, which again somewhat speculative, but is that these kind of form like a suite of traits where the sort of reasons why you might have longer spines, like being more conspicuous, may also correlate with things like having larger colony size or having larger eye size because you're actually above the surface. And it actually might be using your eyes more than like oftentimes blind subterranean species. So that's basically what I would speculate is that there is some kind of functional correlation between these traits potentially, or if not functional, at least yeah. I mean, that would be my guess basically. And it is something that could actually could be studied, especially with more detailed field work as well.
Thank you so much.
Yeah, and thanks for the question. We have another question. It's a Lo. Let me unmute. Hey, Lo.
Hey, Ben, enjoyed your talk, and you're always such a conversationalist in formal talk and also in this online platform. My question is about the tree you manifested the great evolutionary transition back and forth in some of the clades. And my question is really, that's a large scale phylogenetic tree at species level. And you also earlier discussed that you did build some of the ant colonies, and that there are with a number of with a range of size difference in this colony. My question is about how to tie the population level of variation to the macroevolutionary difference on the phylogenetic tree. So what is the largest population that you have where you can do some assessment of variability of the spines?
And when you say variability, you mean like intraspecific variability?
Correct.
Yeah, so one thing that's interesting is that overall, there seems to be fairly low in intraspecific variability, especially relative to interspecific variability. But it would be possible with certain groups like Polyrhacus dives, which have both really super large colonies of like tens of thousands of workers, or potentially even there's a species called Polyrhacus furcata, which is has really long spines. Is one of the groups that has long, it's an independent evolution actually of these long hooked spines. And I could collect enough colonies of those potentially to assess intraspecific variation. And I guess there'd be some interesting questions, yeah, that you could ask there, or a whole number of questions you could ask there. And it would be interesting, like one could ask a question like something like integration or a covariance between traits if that's restricted. This was actually kind of something I was interested in earlier on in my PhD on potential correlations between like spine traits and the general variation within the population of a species. So yeah, I don't know if that fully answered your question.
That's good. I understood.
Oh, cool.
Thanks.
Okay, cool. Thanks for the question.
Great. So if we don't have any further questions, we will now begin the private committee portion of the examination. But I would like to ask everyone, please copy down the Zoom link and the Zoom meeting password so that at 2 o'clock, please log on to that Zoom link to congratulate Benjamin, pending a successful rest of his defense. And Benjamin, thank you again for a great presentation.
Thank you, and thank you so much everyone for joining.